Bonjour tout le monde, merci beaucoup de vous être connectés dans cette troisième conférence de l'année sur la découverte des biomarqueurs dans les maladies mentales et neurodéveloppementales. On a un très très grand speaker aujourd'hui, donc je suis heureuse que vous ayez pu vous connecter. Je pense que ça va être absolument passionnant. Et je vais demander au professeur Anouck Amestoy de Bordeaux d'introduire le professeur Ingrid Tein. Anouck?
Oui, merci beaucoup Marion. Donc bonjour à tous. Donc j'ai effectivement l'immense plaisir de vous introduire le docteur Ingrid Tein qui nous vient de Toronto. Donc le professeur Tein est directrice de la clinique du laboratoire de recherche en neurométabolisme. Elle est aussi neurologue au sein de la division de neurologie à l'hôpital pour enfants malades de Toronto. Elle est professeure émérite, comme on le voit là sur sa diapositive d'introduction en pédiatrie, médecine de laboratoire et pathobiologie. Et elle est surtout associée à de nombreux projets, dont un projet scientifique principal qui est intégré au sein d'un programme de génétique et de biologie du génome. C'est quelqu'un qui a énormément d'expertise sur les biomarqueurs métaboliques dans les pathologies neurodégénératives, mais aussi dans la santé mentale. Donc, elle va nous parler aujourd'hui des potentiels biomarqueurs et des métabolismes anormaux qui sont dans les hypothèses étiologiques du trouble du spectre de l'autisme. Thank you very much, Ingrid. You can start your talk.
Merci beaucoup, Marion et Anouck. Et je commence. I thank you for this kind invitation to present potential biomarkers of abnormal metabolism in autism spectrum disorder. These are my disclosures. Our MITO2 grant from the University of Toronto focuses on genetic susceptibility variants in carnitine metabolism genes in ASD. The goals of my talk are to present an overview of the complex genetic and environmental factors implicated in ASD. Secondly, to identify metabolic etiology of autism and to review potential biomarkers associated with these disorders. And thirdly, to identify specific neurometabolic disorders presenting with neuropsychiatric phenotypes, highlighting the treatable etiologies and their biomarkers. The most important point in all neurometabolic disorders is that the earlier the recognition and specific therapeutic intervention, the better the prognosis. I'll try to set the stage regarding the complex etiopathogenesis of autism, followed by a focus on specific diagnostic groups that have targeted precision medicine therapies, and conclude with a comprehensive big data approach. Autism spectrum disorder, as you know, is characterized by first, problems in social interaction, difficulty in adapting behavior in various social contexts, or lack of interest in peers. Secondly, communication problems, both verbal and nonverbal, such as difficulty making eye contact, facial expressions, body postures, and difficulty in understanding or using the gestures that regulate interaction with others. And thirdly, the presence of restricted or repetitive behaviors, such as rituals that are conducted with a rigid manner. The prevalence of autism is approximately 1% in the general population, with a marked male predominance of 4-1. Intellectual disability occurs in 30 to 50% of cases, and the frequently associated comorbidities include ADHD, impaired motor coordination, gastrointestinal disorders, including chronic abdominal pain, constipation, and diarrhea, with disorders in gut microbiota, and psychiatric disorders such as increased irritability, anxiety, affective disorders, externalization of problems, increased sensory sensitivity, and sleep disorders. The clinical manifestations typically occur in the second to third year of life and usually persist into adulthood, influencing various aspects of mental development. The exact etiopathogenesis of autism is not fully understood, but many studies have investigated both genetic and environmental factors. It is well established that several factors involving the pre, peri, and postnatal environment are associated with an amplified risk. Amongst these, the main factors are maternal infections, stress, and diabetes, in addition to exposure to pesticides, air pollution, infections, inflammatory conditions, and the consumption of antibiotics during pregnancy, which together make up the so-called exposome and combine to increase the ASD risk. Environmental factors can also influence genetic vulnerability by modifying the development of neuronal circuitry. Regulated causes of autism are heterogeneous, involve multiple gene variants found in 60 to 80% of cases with a complex genetic architecture. Approximately 600 to 1,200 genes and genomes have been identified to be associated with autism. At least 5% of ASD cases are caused by single nucleotide polymorphisms, and approximately 10% are due to copy number variants that disrupt protein coding, including chromosomal duplications, large deletions, inversions, and translocations. Many ASD patients have recurrent de novo disruptive mutations, particularly affecting specific protein targets, such as the chromodomain helicase DNA binding protein 8 or CHD8, the activity-dependent neuroprotector homeobox ADNP, and the phosphatase-intensive homolog PTEN. Certain of the high-impact ASD-associated genes involve synaptic formation, such as NRXN1, which affects neuronal circuitry. ASD can be divided into two subtypes, syndromic and non-syndromic. Syndromic refers to mutations in specific genes manifesting with a neurologic syndrome, such as Fragile X syndrome. Non-syndromic or idiopathic accounts for the vast majority and is not associated with other neurologic disorders, but is related to genes associated with autism. However, with the expanding phenotypic spectra, there can be overlap between the two. Rare single gene disorders strongly associated with autism include Rett syndrome in girls, in whom 40% have ASD, and Fragile X syndrome males, in whom 25% have ASD. Interestingly, the majority of point mutations in ASD are paternally inherited, and there's a correlation between paternal age and number of de novo variants, which have a 1.3-fold increase for every 10 years of paternal age. In addition, significant comorbidities that have been reported in the literature in many ASD studies include oxidative stress, inflammation, mitochondrial dysfunction, and immune dysregulation, which are associated with changes in protein and metabolite pathways. Mutations of proteins involved in transcriptional regulation are associated with some syndromes of ASD, including the L-type voltage-sensitive calcium channels in Timothy syndrome, which trigger calcium influx, induction of calcium-dependent signaling molecules, and the RAS-ERK pathways involving transcriptional regulation, the MECP2 gene in Rett syndrome, and the UB3A gene in Angelman syndrome. Mutations of proteins involved in translation regulation include PTEN, ADNP, tuberous sclerosis 1 and 2, and FMRP of Fragile X syndrome, which are highlighted in red in this slide. Intact synaptic structure and homeostasis are fundamental for normal neuronal function and circuitry. Neuropathological studies have provided evidence of increased dendritic spine density and aberrant dendritic spine morphology in individuals with ASD. At the excitatory synapse, mutations in encoded proteins include synaptic scaffold proteins such as the Shanks, neurotransmitter receptors such as the NMDAR and AMPA receptors, and cell adhesion molecules such as NRXNs and NLGNs, which are associated with autism risk. In addition, mutations in ion channels such as the calcium channel and also the sodium channels such as the SCN1A result in synaptic dysfunction and autism-like behavior. Furthermore, there is increasing evidence that ASD is the result of a complex interaction between multiple genes and the environment. In addition, there are also epigenetic factors which are ideally positioned at the genome-environmental interface, which allow for steady gene expression regulation without alterations to the underlying DNA sequence. Epigenetic mechanisms, including DNA methylation, histone modification, chromatin remodeling, and non-coding RNA activity, which are involved in the regulation of social behavior in ASD. Immune dysfunction is another factor contributing to gene-environment interactions in the context of ASD. Persistent immune dysregulation has been identified in ASD patients and animal models. Inflammatory molecular signaling pathways in both the central nervous system and the periphery can affect brain connections and synaptic function by affecting components such as microglia, complement factors, cytokines, and their receptors, and major histocompatibility complex 1 molecules. Dysregulation of cytokines in ASD has been associated with symptom severity and presentation. Recently, the gut has gained attention as a key connection in the microbial immune neuronal system interplay. The abundance of gut microbes in ASD patients, include Clostridium, Bacteroides, etc., is significantly different from healthy controls. Studies in individuals with ASD have revealed that intestinal imbalance can affect peripheral immunologic responses and contribute to immune cell dysfunction. Furthermore, gut dysfunction can affect brain function through neural, hormonal, and immune signaling. Interestingly, the gut microbiota is essential for microglial morphological and functional maturation, and microglial damage can be corrected to some extent by complex microbiota. Therefore, microglia and inflammation alterations in the central nervous system may be at least partially attributable to microbial dysregulation. Twin studies estimate heritability in the 50 to 90% range, and recurrence in families is approximately 20%. Genetic causes are identified in about 20% of families, with 80% being idiopathic. Metabolic factors have been described in up to 10 to 20% of children, including abnormalities in the mitochondria and carnitine-dependent system. Potential pathogenetic mechanisms linking mitochondrial dysfunction and ASD include the mitochondrial handling of calcium, mitochondrial-induced oxidative stress, and abnormal carnitine metabolism. Carnitine is an essential cofactor for fatty acid metabolism. It transfers long-chain fatty acids across the inner mitochondrial membrane for oxidation and ATP generation, shuttles acyl-CoA products between peroxisomes and mitochondria, and acts as a kind of metabolic mop to esterify potentially toxic acyl-CoA metabolites, which may secondarily impair various bioenergetics such as the and fatty acid oxidation during acute clinical crises. But perhaps most importantly, it modulates the acyl-CoA to free-CoA ratio in mammalian cells by complexing to the acyl derivative, thereby liberating free-CoA, which is essential in a number of intracellular energy-generating processes. In carnivores, 75% of carnitine comes from the diet, from red meat and dairy products, and about 25% from endogenous biosynthesis. In vegans, endogenous biosynthesis must be upregulated, or greater than 90% is biosynthesized. As 90% of carnitine stores are found in skeletal muscle, there can be significant tissue carnitine deficiency, but still normal serum concentrations. The normal serum carnitine concentrations are tightly regulated by the renal threshold, which reabsorbs greater than 90% of the filtered load. The mammalian carnitine system consists of 8 different carnitine acyltransferases, which link at least 4 intracellular pools of acyl-CoAs, namely cytosolic, mitochondrial, peroxisomal, and endoplasmic reticulum, that supply a multitude of lipid metabolic pathways. The critical role of the carnitine system is to maintain homeostasis by keeping the acyl-CoA to free-CoA pool constant, even under conditions of very high turnover, and by providing the cell with a critical source of free-CoA. The high-affinity plasma-level carnitine transporter, or octin-2, the intermediate affinity peroxisomal octin-3, and the low-affinity mitochondrial octin-1 facilitate carnitine transport between organelles as in a finely choreographed ballet. Now, we have been interested in the expression of the 3 organic cation carnitine transporters in brain and applied our transporter-specific antibodies, followed by a secondary antibody and DAP peroxidase detection system in brown to serial sections of adult murine brain. All 3 showed strong expression in the olfactory bulb, the arcuate nucleus, which serves in the chemiosmotic sensing of the fuel status of the animal, in the input fibers in the motor cortex, as well as in the cornu ammonis and dentate gyrus of the hippocampus, and in the axons and dendritic interpretations of spinal cord neurons in both ascending and descending white matter tracts, whereas octin-3 was selectively strongly expressed in the anterior horn cells. There was also prominent expression of all 3 in the Purkinje cells of the cerebellum, as shown here, octin-1, octin-2, and octin-3, and in the choroid plexus. Octin-2 is likely of key importance for the exchange of organic cationic drugs in the blood-brain barrier. These patterns of expression are suggestive of the organic cation carnitine transporters in maintaining carnitine homeostasis in neurons, maintaining olfactory, satiety, limbic, memory, motor, and sensory functions. There's a large body of literature on the roles of L-carnitine and acetylcarnitine in the central nervous system for bioenergetic function, generation of free CoA, and in the donation of acetyl groups from acetylcarnitine to choline acetyltransferase for acetylcholine synthesis, as well as in neuromodulatory, neuroprotective, and antioxidant activities. The carnitine balance in the body is dependent on the input side upon the diet, whether it is adequate as in carnivores or low as in vegans, and whether there are any issues with intestinal absorption. It also depends upon the biosynthetic capability of the individual, which is age-dependent, being only 25% of adult levels in infancy, and on the integrity of the high-affinity plasmalomal carnitine transporter OCTIN-2, which is ubiquitously expressed in all tissues and which transports carnitine from the blood into the tissues against a large concentration gradient. Input may also depend upon whether there is any exogenous supplementation of carnitine. On the output side, L-carnitine stores may be reduced by increased utilization or loss, as may occur in conditions of high demand, such as inborn errors of metabolism, diabetes, certain medications such as valproic acid, and renal tubular losses such as in Fanconi syndrome. Now, a defect in the high-affinity plasmalomal carnitine transporter formerly presented with early infantile onset recurrent episodes of hypoketotic hypoglycemic encephalopathy, followed by progressive hypertrophic cardiomyopathy and cardiac failure, and limb-girdle myopathy, which was uniformly lethal in the first few years of life, but which is entirely reversible with lifelong high-dose oral carnitine supplementation with good outcome to late adulthood and good quality of life, provided there be no episodes of severe infantile hypoglycemia. All patients have responded with a dramatic improvement in symptoms within a few weeks and a dramatic reduction in heart size to normal within a few months of therapy. The plasma carnitine concentrations approach normal but are never elevated due to the renal reabsorptive defect. As with many autosomal recessive disorders, the most severe phenotype was first recognized, but subsequently we've identified a milder phenotype in a 6-year-old boy who presented with intellectual disability, ADHD, and ASD features, and who had a highly positive response to carnitine therapy in terms of behavior and development. Now, a reduction in serum carnitine has been shown in 100 children with non-syndromic ASD, in whom there were also slight elevations in lactate and significant elevations in alanine and ammonia in a study by Philippek et al. In 2004. The links between carnitine and ASD rely on 3 lines of evidence. First, alteration of mitochondrial redox function in patients with ASD. Secondly, the relationship between L-carnitine and acylcarnitine levels and the symptoms of ASD with decreased free carnitine and abnormal short and long-chain acylcarnitine profiles. And third, the genetic aspects of ASD associated with carnitine metabolism with single nucleotide polymorphisms and copy number variants in carnitine transporter and biosynthesis genes. 2 small RCTs and 1 open-label prospective trial suggested that carnitine therapy may be associated with improvements in symptoms as assessed by the Childhood Autism Rating Scale and Clinical Global Impression Scales in non-syndromic ASD. The effect of carnitine in ASD associated with genetic disorders is inconclusive due to a lack of proper RCTs, but beneficial effects have been suggested for comorbid intellectual disability and ASD symptoms in mitochondrial complex 2 to 4 deficiencies, propionic acidemia, and defects of carnitine metabolism, including the carnitine transporter OCTIN-2 deficiency, and X-linked trimethylysine hydroxylase epsilon deficiency, which is within the carnitine biosynthesis pathway. That brain carnitine deficiency might lead to autism has been suggested by reports of severe deficiency in ASD and by evidence that X-linked trimethylysine hydroxylase deficiency is a risk factor for ASD, supporting a mixed common gene variant environment hypothesis. Male predominance may also relate to the X-linked SLC6A14, whose inactivation could limit transport of carnitine across the blood-brain barrier. Geneticist Arthur Baudet proposed that 10 to 20% of non-syndromic ASD involving extreme male bias in genetically normal infants may develop due to early brain carnitine deficiency that may be amenable to early reversal and prevention. We hypothesized that brain carnitine deficiency increases the risks of ASD in the subgroup of ASD. Infants have low carnitine biosynthesis rates of 25% of adult levels and are thus dependent upon transplacental carnitine transfer and breast milk carnitine. Infants born to vegan mothers with low tissue carnitine are at high risk for deficiency, and this risk would be amplified by the co-occurrence of mutations in the transporter and biosynthesis genes. The significance lies in the identification of ASD children with dysregulated carnitine metabolism who would benefit from a precision medicine approach with early carnitine therapy in at-risk children, which should improve their clinical phenotype. Furthermore, the earlier the identification of at-risk children and treatment intervention, the greater would be the effect on early brain development, improving social communication, intellectual function, and school performance. So this is an overview of bioenergetic metabolism, in which there are three basic energy substrates, namely fatty acids, carbohydrates, and amino acids, which are metabolized to two carbon fragments of acetyl-CoA, which is then taken down through the Krebs cycle, which generates ATP and reducing equivalents that are then taken down through the final common pathway of the respiratory chain, where oxidation from complexes 1 to 4 combines with phosphorylation at complex 5 to generate ATP. A defect in this pathway, anywhere in this pathway, could lead to energy insufficiency in the brain, affecting neurodevelopment and behavior. Mitochondrial disorders in the aggregate are now recognized as the most common inborn error of metabolism, with an estimated prevalence of up to 1 in 1,000. In children, 80% of mitochondrial disorders are autosomal recessive and arise from nuclear encoded gene defects, with the remaining 20% due to mutations in mitochondrial DNA, which are maternally inherited, whereby the disorders are passed down from the mothers to all of her children, both male and female, but are only transmitted through the female lineage. Mitochondrial dysfunction typically affects tissues with high energy demands, such as brain, muscle, kidney, and liver. They often involve the central nervous system and other organ systems, and may present with encephalopathy, stroke-like episodes, seizures, developmental delay, ataxia, peripheral sensory motor axonopathy, movement disorders, limb girdle weakness with exercise intolerance, as well as ptosis and ophthalmoplegia, optic atrophy, pigmentary retinopathy, sensorineural hearing loss, and hypertrophic cardiomyopathy with conduction blocks, liver failure, Fanconi syndrome, episodes of intestinal conditions, hypoparathyroidism, short stature, and failure to thrive. Given the bioenergetic insufficiency in brain, children with mitochondrial disorders may have intellectual disability, ADHD, and autism symptomatology. There may be an elevation in serum, CSF, and MRS lactate, but normal lactates do not exclude a mitochondrial disorder. So each cell contains many copies of mitochondrial DNA, and during mitotic segregation, the ratio of mutant to wild type will determine the clinical phenotype, with higher morbidity associated with higher percentage mutant heteroplasmy. The most common mitochondrial gene defect is in the nuclear polymerase gamma gene, which is responsible for the integrity and replication of mitochondrial DNA, and which may account for up to 20% of mitochondrial disorders, with mutations that may behave both as dominant and recessive alleles. The recessive mutations present in childhood and tend to cause mitochondrial DNA depletion. The dominant mutations tend to present in adulthood and cause multiple secondary deletions in mitochondrial DNA. Quite remarkably, polymerase gamma mutations can present with almost any clinical mitochondrial phenotype, but importantly, they can present in later adolescence with Parkinsonism and psychosis. I would now like to turn to creatine, which is an important energy substrate in brain, muscle, heart, and bowel, and the creatine deficiency disorders, which have a strong association with ASD and intellectual disability, and which can be diagnosed very early on brain MRS profiles, which show a very low creatine peak at 3 parts per million. I will first review the pathway of creatine biosynthesis, which allows understanding of the diagnostics and the tailored treatments. So arginine and glycine are converted into guanidinoacetic acid and ornithine in the kidney by arginine glycine amidinotransferase. GAA is then transported to the liver, where in the presence of S-adenosine methionine and guanidinoacetate methyltransferase, or GAMT, it is converted into creatine, which is then ready for transport into tissues such as brain, muscle, and heart. The excellent creatine transfer defect affects boys most severely, but can also affect girls who lionize poorly. It accounts for up to 3.5% of boys with intellectual disability. The seizures may be generalized tonic-clonic, focal dyscognitive, or myoclonic, and there's usually severe developmental delay or regression with marked speech delay. There may be an associated extrapyramidal movement disorder. Most notably, these boys have an autistic features and ADHD with major behavioral issues. The unique features include midfacial hypoplasia, as shown here. They also have constipation, megacolon, ulcers, and perforations, which is related to the defective creatine transport in intestinal epithelial cells. They can be diagnosed early based on MRS of the brain, which shows absence of the creatine signal seen in all three creatine deficiency disorders with severe depletion of creatine and phosphocreatine in brain. These children with the X-linked transporter defect have normal or increased creatine in plasma and urine. Most notably, they have normal GAA, which distinguishes them from the other two disorders. Biochemically, there is decreased creatine uptake in fibroblasts. The MRI in these children is mildly abnormal with mildly delayed myelination, a thin corpus callosum, and cerebral and cerebellar atrophy. The MRS profile shows markedly reduced creatine peak at 3 parts per million, as shown here. Please consider including MRS with MRI in young children with developmental delay, autism, and seizures for the very valuable information it may provide. Treatment for the creatine transporter defect includes creatine supplementation. However, this may not be effective depending upon the severity of the transporter defect. The additional, more effective strategy is to promote endogenous creatine biosynthesis in the brain by providing the child with the precursors of L-arginine and glycine. In one study, L-arginine for 9 months didn't show a significant improvement. However, in a longer study of 1 year in a 9-year-old, there was demonstrated improvement in neurologic, language, and behavioral status and evidence of increased brain creatine as shown on this MRS. At present, it is also recommended to give S-adensylmethionine, which combines with the GAA to make creatine through GAMT. The second disorder in this series is autosomal recessive packet deficiency, which has a similar clinical profile and severe brain creatine depletion, in which there is a block in the transfer of a guanidine group from arginine to glycine to form GAA, the precursor of creatine. These children are distinguished by having low blood and urine GAA. There is some improvement with oral creatine, but most importantly, early intervention at 2 months may prevent the phenotypic expression of the disease altogether, which is primary prevention. The third disorder in this series is the autosomal recessive disorder GAMT deficiency. The clinical phenotype is very similar to the other two, but has in addition a marked extrapyramidal movement disorder, including ataxia myoclonus and dystonia, which is likely due to the toxic accumulation of GAA in the globus pallidus, as shown on this T2-weighted MRI. Biochemically, this disorder is distinguished by the accumulation of GAA in brain, which can be seen on MRS at 3.78 parts per million, and in body fluids, which may be responsible for the intractable seizures and the movement disorder. The pathology is characterized by marked myelination delay. The treatment with oral creatine is only partly successful. In this MRS, you can appreciate the increase in brain creatine on MRS following creatine therapy. The clinical outcome is improved by restricting dietary arginine and providing ornithine in order to reduce the production of GAA. Treatment at the asymptomatic stage actually prevents the phenotype. The earlier the introduction of therapy, the better the outcome, though even in young adults that have been missed, there can be a clinically significant response to therapy of the movement disorder by reduction of the GAA. We will now focus on pyridoxine and pyridoxal L-phosphate responsive seizures and encephalopathies, which may present with autism, and focus on 2 of these disorders out of the 7. Pyridoxine, or vitamin B6, is converted to its biologically active form of pyridoxal 5'-phosphate, or pyridoxal L-phosphate, PLP, by the enzyme PNPO. Pyridoxine has many cellular functions, including balancing sodium and potassium, decreasing the formation of homocysteine, and is a precursor for PLP, which is the critical cofactor for aromatic amino acid decarboxylase, which converts 5-hydroxytryptophan into serotonin and L-dopa into dopamine, noradrenaline, and adrenaline. The dietary sources include dragon fruit, grains, and nuts. This is the dragon fruit from Southeast Asia. Pyridoxine deficiency may lead to cheilitis, as shown here, and conjunctivitis, and sideroblastic anemia, as well as neonatal onset seizures, irritability, and confusion. Pathophysiology of the CNS phenotype likely relates to the impairment of the decarboxylation of glutamate to GABA, which is important in inhibitory neurotransmission, and the impaired transamination of glutamate to alpha-ketoglutarate, which is critical for mitochondrial anaplerosis and ATP generation. In contrast to the deficiency disorder, we have the genetic autosomal recessive disorder of pyridoxine-dependent epilepsy, which is due to alpha-aminoadipic-semialdehyde dehydrogenase deficiency, which is encoded by the antiquidin gene. The prevalence of this disorder varies from 1 in 20,000 to 1 in 64,000 and is seen in all ethnic groups. Depending upon the severity of the mutation, one may have prenatal onset with intrauterine seizures or postnatal seizures that begin on the first day of life. However, remarkably, there have been milder cases that have presented as focal seizures as late as 3 years of age. The seizures may be intractable, clonic, generalized tonic, myoclonic, or rarely infantile spasms. In the classic severe cases, the infants are resistant to all standard anti-epileptic drugs and have complete and immediate cessation of seizures with intravenous pyridoxine. Additional features may include respiratory distress, acidosis, abdominal distension with vomiting, and poor sleep. Despite early therapy and good seizure control, most children will have mild to severe developmental delay and speech delay, though a small proportion have had normal development. MRI may be normal or show hyperplasia of the corpus callosum, ventriculomegaly, and atrophy. It's been suggested that prenatal therapy with maternal pyridoxine may improve outcome. However, this could only happen for a subsequent sibling in a family following prenatal testing. The defective enzyme is alpha-aminoadipic semialdehyde dehydrogenase in the piperacolic acid pathway of lysine catabolism. The biomarkers are an increase in plasma, urine, and CSF piperacolic acid and AASA. The pathophysiology relates to the production of piperidine 6-carboxylate, which inactivates the PLP. The behavioral and psychiatric phenotypes include autism, anxiety, ADHD, hyperactivity, obsessive compulsive disorder, and mood disturbances. Additional biomarkers include serum hypoglycemia, lactic acidosis, raised threonine, glycine, taurine, histidine, and 3-methoxytyrosine, and in the CSF, low PLP and GABA, and raised glutamate, threonine, glycine, taurine, histidine, and 3-methoxytyrosine. The biochemical pathophysiology of antiquidin deficiency is shown on this slide. So L-lysine is catabolized by the saccharopine pathway in mitochondria to AASA, and by the piperacolic acid pathway in peroxisomes to piperidine, which is equivalent by the AASA. Both accumulate proximal to the block in the enzyme, and the piperidine 6-carboxylate undergoes chemical condensation with PLP, resulting in PLP deficiency. The piperacolic acid accumulates due to back pressure from the enzymatic block. This slide demonstrates the urinary AASA concentrations in patients here in the dotted lines compared to the control populations. You'll note that there's an age-related decrease in AASA. However, the patients have consistently elevated levels, and these persist despite treatment with pyridoxine. Our second genetic disorder in this group relates to a block in pyridoxamine 5'-phosphate oxidase, or PNPO, which converts pyridoxine into PLP. They may have premature birth and seizure onset day 1, or even in utero. There are now 3 subgroups proposed based on the severity of the mutations. Neonatal onset seizures, which may be clonic, myoclonic, or status epilepticus. These children are distinguished by their rotatory eye movements, hyperexcitability, and hypersalivation, and their EEGs may show a severe burst suppression pattern or myoclonic epilepsy. These children respond to PLP. Secondly, infantile spasms, which respond to PLP. And thirdly, infants with seizures starting before 3 months of age, which respond to pyridoxine. The biochemical markers include hypoglycemia, early acidosis, pancytopenia, and coagulopathy. The CSF and urine biochemical profile is consistent with the reduction in PLP-dependent enzymes, including aromatic amino acid decarboxylase, which results in a decrease in CSF HbA and 5-HIAA. Quantitative amino acids in the serum demonstrate raised glycine, threonine, taurine, and histidine, and low arginine levels. This slide demonstrates the pathway for conversion of pyridoxamine phosphate and pyridoxine phosphate by PNPO into pyridoxal L-phosphate. This slide demonstrates the pivotal role of PLP as the cofactor for aromatic amino acid decarboxylase in the conversion of L-dopa into dopamine, norepinephrine, and epinephrine, and 5-hydroxytryptophan into serotonin. The resultant deficiencies in dopamine may contribute to the dystonia, intellectual disability, and ASD, and in serotonin to the sleep disturbances, restlessness, and irritability. associated with normal neurodevelopment in childhood up to 11 years of age with normal neuroimaging as reported from Australia. It is possible that paradox and responsiveness in PNPO deficiency is affected by prematurity and age at the time of the therapeutic trial. Other additional factors that are likely to influence treatment response and outcome include riboflavin status and how well the fetus has been supplied with B6 by the mother. I will now focus on the glucose transporter or GLUT1 defect, which may have associated autism. GLUT1 may present with three phenotypes, which relate directly again to the severity of the mutation. Classic type 1 phenotype presents with early onset seizures with encephalopathy, usually after the closure of the blood-brain barrier, acquired and progressive microcephaly, developmental delay, and a mixture of pyramidal, extrapyramidal, and cerebellar signs. This is the severest form, which was first recognized, as occurs in most genetic disorders. They may also manifest with behavioral abnormalities in autism spectrum disorder. Subsequently, milder mutations were found, which presented primarily as movement disorders, which now form the majority of known cases. Type 2 form presents with dysarthria, dystonia, and ataxia, and type 3 form is highlighted by choreoathetosis, paroxysmal eye and hip movements, or paroxysmal exertional dyskinesia and seizures. The seizures are variegated and may include generalized tonic-clonic, myoclonic, atypo-absence, and atonic seizures, and the EEG may show generalized spike or polyspike and wave discharges, mild slowing or focal discharges. These children also have marked sleep disturbances and headaches. The cardinal biochemical signature is CSF hypoglycemia, in which the CSF to blood glucose ratio was originally considered to be less than 0.4 in the absence of CNS infection. However, now with milder cases, this has been liberalized to less than 0.5. These children also have a low CSF lactate. Biochemically, there is low red cell glucose transport. This slide highlights the fact that there are a family of glucose transporters in the central nervous system. You'll note that GLUT1 is expressed in granule and pyramidal neurons in astrocytes as well as in the blood-brain barrier. Now, this boy has GLUT1-associated dystonia. The MRI may be normal or show mild enlargement of the CSF spaces, mild myelination delay, and or high T2 signal of subcortical U fibers. PET scanning shows a global decrease in glucose uptake in the motor cortex, mesial temporal regions, and in the thalami. The pathology relates to an impairment in blood-brain glucose transport. It must be remembered that the child's brain has a 3 to 4-fold higher requirement for glucose than the adult brain due to the rapid growth of the brain to 75% of adult size in the first 3 years of life, as well as to the higher metabolic rate of the brain up until 8 years of age during highly active synaptogenesis and dendritic barbarization. Treatment is through the use of the ketogenic diet, which has been instituted as early as 6 weeks of age by Dr. Jorge Klepper at a time when there is sufficiently robust ketogenesis through the fatty acid oxidation pathway in infants. This may be accomplished through the medium chain or long chain triglyceride oil diet, during which the child needs to be monitored for renal stones and other side effects. This diet provides good control of seizures and motor symptoms, but has a lesser effect on cognition, as ketones provide an alternate energy fuel for the brain, but do not serve the signaling role of glucose in the brain. GLUT1 is inherited primarily by autosomal dominant transmission and rarely autosomal recessive, and mutations are identified in the SLC2A1 gene. This graph highlights the dramatic response to the ketogenic diet in a girl who had deceleration of brain growth in the first year of life to 3rd percentile, and following institution of the diet had marked catch-up growth to the 50th percentile. Once again, the earlier the recognition, the better the clinical outcome. There is a large group of inherited neurotransmitter disorders which may present with depression, irritability, and sleep disturbance. Their clinical phenotype will include congenital onset with hypotonia and developmental delay. They may have a variegate movement disorder, including dystonia, truncal hypotonia, oculogyrate crises, bulbar dysfunction, or Parkinsonian features, and they often have autonomic dysregulation characterized by excessive sweating, temperature dysregulation, ptosis, meiosis, hypoglycemia, and irritability. Now, this slide demonstrates the classification of the inherited disorders of biogenic amines into 1, primary disorders of biogenic amine metabolism, which can be divided into disorders of biosynthesis, catabolism, and transport. Secondly, disorders of tetrahydrobiopterin metabolism, which can be divided into disorders of biosynthesis and recycling. And thirdly, co-chaperone-associated disorders, for which time does not permit a detailed review. So this slide demonstrates the specific biochemical metabolite patterns in plasma, dried blood spots, urine, and CSF that can differentiate between various neurotransmitter defects based on normal range, above range, and below range values in plasma phenylalanine, prolactin, serotonin, 3-O-methyl-dopa, dopamine, and catecholamine levels, and in urine, permapterin, neopterin, biopterin, sepiapterin, HVA, phenylactic acid, 3-methoxytyrosine, norepinephrine, and phenylethylamine levels, and in CSF, tetrahydrobiopterin, HVA, 5-HIAA, 7-biopterin, 5-methyltetrahydrofolate, sepiapterin, BH2, and with the HVA to 5-HIAA ratio, L-dopa, norepinephrine, and epinephrine. Now, a disorder for which we have recently published an important interventional therapy is beta-catenin deficiency due to mutations in the CTNNB1 gene, which is a defect in dopaminergic neurogenesis and in the regulation of the canonical WNT signaling pathway that presents with ASD, ADHD, severe anxiety, behavioral problems with self-injury, and tics and sleep disturbance. This is an autosomal dominant disorder characterized by distinct facial dysmorphism, speech delay, motor delay, intellectual disability, microcephaly, truncal hypotonia, and peripheral hypertonia as spasticity and dystonia, which is often overlooked. We studied a 3.5-year-old girl who presented with all of the above features with marked speech delay, severe anxiety disorder, and mixed spasticity with dystonia, who could only walk a distance of 4.5 meters on a 6-minute walk test with a walker, bilateral leg flows, and parents on either side. Following titration with levodopa up to 8 milligrams per kilogram per day, she was able to walk 286 meters independently. Her fine motor and language and cognitive delays improved to age-appropriate skills by 5 years of age. Reduction in the earlier the diagnosis and intervention, the greater the therapeutic effect during critical periods of development. Now, given the large number of genes implicated in ASD, as well as the numerous modifying environmental factors, an integrative approach could be a new strategy for the deep profiling of ASD, which combines data from genome sequencing, such as next-generation sequencing, with those from proteomics and metabolomics by 1 or 2-dimensional gel electrophoresis, liquid chromatography, and mass spectrometry, or even from metabolomics data obtained by nuclear magnetic resonance experiments. The strength of a multi-omics approach resides in their ability to provide complete profiles of biologic features in genes, transcripts, proteins, and metabolites to obtain a broad description of a biological system by systems biology. A non-targeted and high-throughput search of the genetic scaffold of DNA and functional reservoir in RNA, proteins, and metabolites is key to decoding the pathophysiology of complex biologic systems, providing the most appropriate stratification of the disease or identification of novel biomarkers. Now, Ristori and colleagues in 2020 conducted a review of the literature to evaluate the role of metabolomics data for disease phenotype stratification and biomarker discovery in ASD patients. They conducted a PubMed search of papers from 2004 to 2019 using the terms autism, metabolomics, metabolites, or omics. They included observational, prospective, and retrospective studies, case control studies, cohort studies, or systematic reviews, and studies involving metabolomics-based biomarker research in ASD. The aim was to identify new potential biomarkers that could be interpreted for appropriate evaluation in decision support systems. They analyzed 119 metabolites and generated a table of metabolomics-based targets of ASD with the tissue of expression, brain, blood, urine, number of ASD subjects and controls, the analytic technique used, and the metabolite implicated. The relevant metabolic processes included nucleotide metabolism and cell growth and proliferation, purines, pyruvate, propionate, Krebs cycle, galactose, sucrose, nicotinamide, cysteine and methionine, arginine and proline metabolism, energy production, including fatty acid beta oxidation and mitochondrial dysfunction, endocannabinoid metabolism, xenobiotics, the urea cycle, and phospholipid and sphingolipid metabolism, as well as glutathione and oxidative stress markers, mammalian microbial co-metabolism, dopaminergic and serotonergic pathways, synapses, tryptophan metabolism, gut microbiota, and gastrointestinal dysfunction. They then analyzed the metabolite data for the biological process and clustered the metabolites into a number of categories, including lipid metabolism, glycine and serine metabolism, tryptophan metabolism, transcription translation, histidine metabolism, glutamate metabolism, thioguanine action pathway, tyrosine metabolism, glutathione metabolism, nicotinate and nicotinamide metabolism, galactose metabolism, and glutaminolysis in cancer. They concluded that the altered metabolites were mainly associated with fatty acid metabolism, oxidative stress, mitochondrial dysfunction, amino acid metabolism, cholesterol metabolism, energy generation, intestinal microbiota, and neurotransmitter defects. They then developed a clinical decision support system to screen and improve age of diagnosis in children with ASD. In box A, the flowchart included all data use from surveys, multiomics data, and electronic medical records, which were fed into an artificial intelligence machine learning model to classify patients. Once the model had been constructed with good accuracy, the clinician in box B would enter the specific data related to the patient's features, such as high functioning, low functioning, or control groups. They speculated that the system might be used by clinicians to prove early diagnosis because it provided significant information about the features of the ASD patient. This is the big data approach, which will be essential for the early detection of ASD and the possibility of optimizing the pathway of diagnosis, therapeutic intervention, and prognosis. These biomarkers may improve diagnostic accuracy, ASD population stratification, and the development of effective personalized treatment. However, this must all be directed by the clinician's expertise. We must not lose the clinician. On this slide, I've listed potential biomarkers in ASD, summarized from the blood, urine, CSF, and MRS as a beginning. To conclude, ASD is a multifactorial disease that includes multiple genetic and environmental factors that may interact. The phenotype of ASD includes abnormal neurodevelopment, which may be related to genes affecting neuronal synapses and cortical pathways with potential alteration in neurotransmitters. Furthermore, the ASD phenotype may be characterized by mitochondrial dysfunction, oxidative stress, neuroinflammation, and abnormal immune regulation. All of these dysfunctions produce possible biomarkers that could be identified by a multi-omics approach to study ASD. And I'd like to leave you with this final slide of Sir William Osler, who very prophetically stated that the value of experience is not in seeing much, but in seeing wisely. I thank you so much for your kind attention. I left a series of references here for those who would like to pursue further reading in this area. Thank you.
Thank you so much, Ingrid. It was an amazing review of all your knowledge. I think it's a lifelong review, I think, of knowledge and findings. Not so sure we can absorb so much, but you agree we can circulate your slides to those of you who are interested, in particular to the network of on autism. I think that would be important for them to slowly reread and re-screen your slides and references. So extremely rich. I will pass on the floor to Anouck to...
Yeah, thank you. I confirm it's a very complete and detailed presentation. Thank you for this overview. It's very exciting. First, we will try to take some questions from participants. Do someone wants to take first the and speak in French or in English? Yeah, Bruno is...
Many thanks for your wonderful presentation. I just have a short question. In one of your slides, you write the critical question of reduce BH4 biosynthesis. And do you know if, in fact, trial with L-methylfolate, where L-methylfolate is able to modulate BH4 metabolism, were conducted in ASD patient with beneficial effect in this clinical condition?
So you're asking about the use of methyl tetrahydrofolate in
Exactly.
BH4 metabolism defects.
Exactly.
It is certainly something that has been proposed and used. I don't have myself personal experience. I only know what's out there in the literature. People do support providing that in order to what should I say, stimulate the pathway. There are oftentimes many cofactors that are important. And I have to say that I've been extremely impressed with the role of pyridoxine and pyridoxal L-phosphate, which seem to be involved in about 80 pathways, not just the specific genetic ones that we've recognized, but they seem to modulate a number of different pathways. So I think that if one has a deep understanding of where the defect is occurring and what that pathophysiological pathway is and what the outcome is, then one can develop strategies to either, let's say, supplement the pathway if it's a transporter defect, for example, to push through by giving high doses of the precursor. If there's still residual activity, sometimes that's a strategy that works. Alternatively, one can try to bypass the pathway in bioenergetics by giving an alternative fuel. For example, with the GLUT1 transporter defect, giving the ketogenesis pathway as an alternative fuel, though it doesn't subserve all of the functions. And thirdly, there may be additional cofactors that help to stimulate downstream effects. So I think that the more exquisite is the understanding and analysis of the pathway, the more possibility there is for targeted specific therapies that can stimulate and, let's say, modulate the pathway. So certainly.
Thanks. Many thanks.
Okay, so I will have a plethora of questions, but I'm going to focus on some of them. I was particularly interested by the fact that you illustrated a lot of neurogenetic conditions in child, not specific for with autism, but you illustrate that there is a lot of sleep disorders or disturbances in these features. Associated with metabolic dysfunction, various metabolic dysfunction. Are there any elements suggesting a link between mitochondrial or metabolic dysfunction and the physiological pathway of melatonin? Because we know that in ASD, melatonin is one of the answer and a good answer to sleep disturbance in children.
Mm-hmm.
So
yes, I think particularly for those disorders that result in an impairment of serotonin biosynthesis. So if one consider any pathway, so we go from 5-hydroxytryptophan as the precursor, and then that enzyme, aromatic amino acid decarboxylase, which has a pyridoxal L-phosphate as its cofactor, that's what converts 5-hydroxytryptophan into serotonin, and then 5-HIAA. And any pathway that modulates or reduces serotonin, it seems to be a common theme that children with serotonin deficiency will present with sleeplessness and marked irritability. And then that ends up contributing, I think, further to the phenotype, because as you know, sleep is incredibly important. And in a normal sleep cycle, we go through stages 1, 2, 3, 4, and REM over an hour and a half, and we have about 5 of those cycles per night. And those are extremely important for 2 reasons, and probably many others, but particularly for memory. Being able to review the learning of the day before and to be able to consolidate those memories into short and long-term memories. So memory consolidation is very importantly modulated by having those good deep sleep cycles. And secondly, what we've been learning over the past 15 years, I would say, is that the brain has an interesting lymphatic system. So brain has lymph, and this is a very new area, and they're glial lymph, and it drains most of the sort of toxins that accumulate, or the biological end products that need to be eliminated are drained by that lymphatic system. And the great majority of lymphatic drainage occurs during sleep. So if you're not sleeping properly, then number one, there's irritability. Number two, there's a problem with memory consolidation. And number three, you're not adequately draining those toxins or those end metabolites that have been degraded away from the brain. So you can appreciate that environment becomes very, let's say, unfavorable to neurodevelopment and speech development. So I think strategies to try to improve sleep are extremely important for everyone, but especially in these children with autism disorder, because irritability is an extremely important aspect, as well as the sort of hyperactivity and attention deficit disorder that can be associated.
Yeah, absolutely. You're absolutely right. I can go on these aspects because I had the impression, but perhaps I'm wrong, that the clinical trials that you was referring to on the basis of ASD, I mean, focused their targets on global social competencies or cognitive functions, but not not a lot on some other symptoms that can be linked more clearly to metabolic physiopathology. And I was thinking of gastrointestinal dysfunction, but sleep disturbance as well. And this dimension that you mentioned just now, irritability in ASD children, all these kind of dimensional symptoms. And I was wondering if you promote to focus on alternative targets for future clinical trials.
So alternative targets in terms of?
Like sleep symptoms, like gastrointestinal symptoms, or you mentioned the big place of autonomic neurologic pathway, all these kind of associated symptoms that are not the core features of autism.
Yeah, I think the more one has a deeper understanding of a particular child's phenotype and is able to break it down into different units, for example, gastrointestinal disturbance, which may be a significant part. I was fascinated by that link between microbiota of the intestines and the formation of microglial cells, which are very important in support mechanisms within the brain. So I think that with every child, it would be helpful, to break down each one of the specific symptoms that are mostly troublesome and target therapies that are specific to those symptoms, in the aggregate. If one has a problem with, let's say, synaptic formation, well, that's a very, very difficult sort of area to address. But if the other components involve sleep disturbance, then I think one can certainly target trying to manage the sleep disorder. If there is significant gastrointestinal complaints, we know that these children have very different microbiota. They have a large range of different sort of bacteria that have been identified. And in mouse models, by creating some of those, they've been able to recreate some of the intestinal issues and problems, on a murine model level. So people have tried with different sort of probiotic strategies to improve that intestinal flora and to see whether that then helps the child from a clinical standpoint. But one of the things that I would highlight is I know that there are a number of exclusion diets that people try out there. And my major concern there is that it's paramount that the child's nutritional status be carefully monitored, because one does not want to create nutritional deficiencies, because that can then further exacerbate the neurologic condition. And as an example, I'll give a couple of very simple examples. Iron deficiency anemia is extremely serious because it certainly affects neurodevelopment and the neurodevelopmental trajectory because you're decreasing your oxygen carrying capacity. Iron deficiency also can cause significant sleep disturbance because of restless leg syndrome, which can basically keep a child up the entire night. Other deficiencies that one must really look for are B12 deficiency. With B12 deficiency, one can have dementia and subacute combined degeneration of the spinal cord and peripheral neuropathies. Folate deficiency, another one, if you have insufficient folate, that also can lead to a dementia sort of phenotype. So I think that if one is manipulating the diet, it's extremely important to make sure that the child is getting all of the important vitamins and minerals and nutrients that they require, because one can cause secondary dysfunction because of causing these deficiencies. So whatever diets are tried, this is my most important point. Please look at, all the trace elements and nutrients and the important vitamins that the child should be receiving. If they become pyridoxine deficient, well, then they can. So, one wants to be very careful about that.
Yeah, you're absolutely right. I mean, it's a very big issue in France, but it's there's a big alert in each national recommendation. So we hope this perspective, because I think it's a perspective too, will be controlled and secure for all of them.
Yeah, and other things I've mentioned is docosahexaenoic acid, a very, very important, very long chain essential fatty acid, which is very important in the retina. 60% of your phospholipid layer is due to DHA, so one can get pigmentary retinopathy if you don't have adequate DHA stores. And in addition, it's important at synapses. So again, that's very critical for neuronal function. So we must be very cognizant that we are not causing secondary deficiencies. And the last one I'll mention is vitamin D. So vitamin D is not only important for bones, but it's also very important in the immune system. And I've mentioned a couple of times that many of these studies have looked at immune dysregulation in autism. So once again, one doesn't want to enter the child into a secondary deficiency state that then can cause symptomatology and can further exacerbate their complex underlying pathophysiology. So optimizing, their nutritional environment, I think is very important for their development.
Thank you very much. This is a good alert at the end of this very, very nice presentation. Everybody are sure that they don't want to capture Ingrid Tein for more seconds or minutes, because are you okay for sending your presentation for all of us to have the time to slowly look
Absolutely.
again?
Absolutely. And also, I did leave a list of references at the end. So, that talk about a lot of these biomarker profiles. So I realized that it was a great deal of information and very fast, but it's an enormous topic. And I tried to really highlight those disorders that have autism as a very strong association, but give us unique windows of insight into potential pathophysiologic mechanisms. And that's where inborn errors of metabolism have been really, I should say, very striking in providing us with information for potential new precision medicine therapies. Because if we can understand that pathophysiology, that we may then have tools that can be specific it and can at least improve certain aspects of the typic expression in autism.
This is exactly what you have defended with us today, and thank you very much for that, because it's a really big challenge in autism, the question and the issue of the heterogeneity of this disorder. So we have to really take into account all of the pathways, and this is This is
Yeah, every child is unique. Every child is unique.
exactly.
And as they say, we're all zebras. We all have our own unique genetic and environmental milieu. And the more we can understand specific pathways that can cause specific dysfunctions, the more we will have tools to remedy them. And I must say that dopamine is an extremely important neurotransmitter for development. And we learned a great deal about that from our beta-catenin deficiency patients. Once we correct their dopamine deficiency, especially if it's very early in their life, if it's later, we don't have quite that same effect because we've missed the developmental window for language development. But if we can get in early diagnosis and early intervention, we can have quite a significant impact. And that's why I presented that beta-catenin deficiency case, because it was remarkable how her language, which was very delayed, she had very few words at 3.5, but by 5, she was being age appropriate. And also the anxiety in that girl. When she was in clinic, she would not permit me to look at her. If I even looked in her direction, she would start screaming. And initially, my exam was through the father. The father, I asked him to do various things with his daughter so I could observe her. And when we were able to get her up to that maximum dose of dopamine, she came into the clinic and she allowed me to do a full exam. And now she comes into clinic and she hugs me. It was a complete transformation of the personality from severe anxiety disorder to now being very socially interactive and very loving. So I must say, I was very impressed with the transformation of that particular girl, but we were able to treat her very early. My other patients that were diagnosed later and sent to me have not had quite the same response in terms of their development. Their movement disorder, yes, but not so much as in their development, because that critical window of time.
That's past. Yeah.
Yeah.
Okay. And it
So,
was
so
very
that's why I always emphasize early recognition and early diagnosis. And boy, if you do an MRI, do an MRS, because
yeah.
that can tell you lactate elevation, maybe mitochondrial, low creatine, maybe one of those three disorders, and X-linked creatine deficiency being responsible for up to 3% of males with intellectual disability is quite high.
Yeah.
And also the MRS for guanidinoacetic acid, which tells you that they have GAMP deficiency. You know, it's a very simple addition. And I hope that we will be able to see more metabolites in the future, because people are working very hard on using 3 Tesla MRI. No, not 3, 7 Tesla MRI to identify even more peaks. So anything we can do in a non-invasive way would be very pivotal, I think.
You have a very optimistic position. I think we should have this one too, because in France, not all children that are diagnosed, even if it's early in the developmental trajectories, they don't have all MRI, just MRI. So we have to move forward for sure. Thank you again very much. Hope it's a beginning of a sort of perspective of other exchange and other discussions for us with you. And thank you again. Everybody's okay to quit. Okay. Thank you again, Ingrid.
It's my great pleasure.